Dear student, today we will start with biotechnology principles and processes. Now, biotechnology is not something which is new. Humans have been using microorganisms from thousands of years. Louis Pasteur, for the first time, described fermentation as a biochemical process in which the enzymes from yeast were responsible for anaerobic respiration. So, what he could observe that malt is converted to beer in presence of yeast, and then later on he found out what was the biochemistry behind. So the sugar which is present in the malt is converted into alcohol, and this reaction is described as fermentation, or what we called as anaerobic respiration. Okay, now here the enzyme from the yeast, that is zymase. Okay, so not the yeast, he added the extract of yeast later on, and he found that the sugar is converted into alcohol. Then later on it was found out that there were enzymes which were present in the yeast extract, and these enzymes carried out this conversion. So here the use of natural capability of organism was exploited. Okay, so if you use the yeast, or you use the extract, you'll get alcohol from the malt. Now let's talk about the modern biotechnology. So called as the defined definition of modern biotechnology is: it is the branch of biology that deals with application of scientific and engineering principles to the processing of material by biological agent to provide goods and services. Now I'll break down this definition to make you understand what it tells you about. Okay, so here we are talking about the application of a scientific and engineering principle. So that is nothing but it involves the technology. Now what kind of technology are we using in biotechnology, such as tissue culture, protoplasm fusion, immobilization of enzyme, genetic engineering, cell catalysis, and many more techniques are there. So here we are using these techniques since they are based on certain scientific principle. Okay, to processing of material, basically here we work with the DNA, because we know that the entire information of the organism is encoded in DNA by biological agent. So biological agent could be the whole organism, could be the part of organism, could be any biological product, could be any cellular component, or biomolecular processes such as transcription, translation, what we have done with. Okay, so any of this biological agent is used in the technique by biological agent to provide goods and services. So goods and services means they are useful to mankind, maybe in the form of DNA vaccine, maybe in the form of gene therapy, maybe in the form of artificial insulin. So let's see what is the definition. It is the branch of biology that deals with application of scientific and engineering principles to the processing of a material by biological agent to provide goods and services. Okay. Now next we will be dealing with certain important terms which will help you to understand the biotechnology. So first we are going through these terms. First is the genetic engineering, which is also known as gene manipulation. Now it is the process of using recombinant DNA, that is rDNA technology, to alter the genetic makeup of the organism. Now whatever is the original genetic makeup which is present in the DNA is called the genetic makeup of the organism. If you alter the DNA, the genetic makeup of the organism is altered. So this process is called gene manipulation or genetic engineering. Now recombinant DNA technology, what is that? It is the technique used to produce recombinant DNA that can be introduced in host to produce desired product. Let's see what is recombinant DNA, which is also known as transgenic DNA, chimeric DNA, rDNA. The DNA from two different sources are joined to create hybrid DNA. This is called rDNA or recombinant DNA. so that it can be inserted into host to produce new genetic combinations. Okay. Now, transgenic organism, we also call them as genetically modified organism or GMO. These are the organism whose genetic material has been altered using genetic engineering, and it will express modified phenotype. Cloning, let's see what is cloning. We know that it is the process of generating genetically identical copy of cell or organism. Usually during the asexual reproduction, all the clones are produced. But here we'll be talking about DNA cloning, which is the process of making multiple copies of particular piece of DNA, maybe by replicating the organism. So these terms are actually interchangeably used. Now we will be discussing one example in which I will be explaining all these terms so that these terms are clear to you and you know how to use them. So let's take the example of BT cotton plant. Now BT stands for Bacillus thuringiensis. It is a kind of gram positive, you can say, bacteria. Cotton plant, you know. Now BT cotton plant is used for the genetically modified cotton plant. So what is BT cotton plant? It is genetically modified plant which is resistant to pest, as it is able to produce insecticidal proteins. So what is the pest which is affecting the cotton plant? It is the bollworm. Bollworm is actually the larva form of moth. So this is the pest which was affecting the cotton plant. And since this genetically modified plant is there, it is able to produce the insecticidal protein. So this is what is the modified phenotype of this genetically modified plant. Since we are making changes in DNA, so the phenotype is automatically altered. If you see normal, if you compare this BT cotton plant with the normal cotton plant, this is susceptible to bollworm. Bollworm is a insect which is you can say which affects the cotton fruit, and as a result what will happen, there is a loss of crop yield. So we want to control this bollworm. We cannot use so many insecticide because the use of chemicals in the you can say agriculture is harmful to the environment. So this is the alternate which is produced with the help of biotechnology, or you can say recombinant DNA technology. Now right now I'll be discussing in very simple terms how this is produced. I'm not going in details of the biotechnological terms because we will discuss that later on. Here you must understand what is the recombinant DNA technology in common terms. Okay. So how this GMO is produced by recombinant DNA technology? The first step is isolation of insecticidal protein. Now we know that Bacillus thuringiensis is the bacteria which produces certain toxin proteins which are insecticidal to the bollworm. So these protein, or we can say which are the toxin of these bacteria, first, since it is produced extracellularly, so you can isolate this protein. So first step is the isolation of insecticidal protein from Bacillus thuringiensis, and this protein is called cry protein. uh, we will say this is a gene product. Since this is a protein, so this is a gene product, and this is cry comes from the crystalline protein. The protein is toxic to bollworm. So we are interested in isolating this protein. So this is done. Here certain techniques are used to isolate the protein. We will see in the chapter which are these techniques which help in the isolation of the protein. The next step is isolation of DNA. Now we know that each protein is coded by DNA, certain DNA, or you can say gene. Okay, and this gene is present on the DNA. So We will now try to locate what is the DNA of cry protein. So for that we have to now isolate the DNA. We will isolate the DNA of Bacillus thuringiensis, and then from the DNA we are going to isolate this particular gene called cry protein. Now there are again many techniques which are involved in this process, how from the protein product we isolate the gene. So now once we got this cry gene, okay, these there are two cry genes for this, cry 1AC and cry 2AB. Now they produces together the cry proteins. These genes, now what you will do, these genes are to be introduced in the cotton plant. So where you will introduce in the cotton seed. Now introducing this gene in the cotton seed is the next step. Now this cry gene is our desired gene, you can say, which is to be introduced in the cotton. So cotton, you can see cotton seed act as a host, whereas cry gene is a desired gene, or we can say this is also foreign gene because it is foreign to cotton seed. Okay, so for this extra vehicle is required. Now there are many methods to introduce gene into the host cell. We are going to study about all these methods. Okay, so one of the method is that we form the rDNA, that is the cry gene is loaded to vector DNA, and this from the recombinant DNA, which is again this recombinant DNA carry the foreign DNA, and this is now introduced into the cotton seed. So next step is the genome of host, that is cotton seed, is now, once it is introduced, it will go and integrate into the cotton seed. So now the cotton seed carry foreign DNA, that is cry gene, along with its own DNA. Now this will get expressed in the host, that is in the cotton plant. So such kind of modified seed are called transgenic seeds. Okay, and the plant which is produced from this is will be genetically modified, because it will be expressing what, genetically modified phenotype. Earlier this normal plant was susceptible to bollworm. Now since this GMO plant is able to produce the insecticidal protein, that is cry protein, and this protein will provide the resistance to the pest to the plant. So we say this is genetically modified cotton plant. So this is just one example to show you what is recombinant DNA technology. There are so many technological things which are involved at each step, and then we are going to study about them. So next we will be dealing with the tools of biotechnology. Thank you.